Good morning, good evening, good afternoon from wherever you're joining in, and welcome to this exciting session today. And to be honest, if a few decades ago you told someone that one day you could identify the species, sex, and even individual of a species of interest with the help of hair or feces, they probably hardly believe you. In fact, what used to be science fiction is today a reality, and it's people like Dr. Meng Yao, Dr. Cheng Chen, Dr. Umar Ramakrishnan, Dr. Byron Weckworth, Dr. Kozuki Noshita, and many others who are making it so. So from identifying individuals to estimate populations to decoding diet, from assessing the level of stress animals are undergoing to finding out where an illegally traded skin is coming from. Conservation genetics today has equipped ecologists and conservationists with some of the most formidable and reliable tools. And the genetic methods and their applications are evolving rapidly, and developments in environmental DNA, conservation genomics, hormone analysis. And what we're going to hear about is I think, wasn't it barcoding, some sort of barcoding? They're offering possibilities to understand and conserve threatened wildlife. So as I started with saying, I know as much about genetics as perhaps my 7-year-old daughter, but it is experts such as Dr. Yao and Dr. Chen who help us decipher genetics and make it accessible and replicable for conservationists. So without much ado, I'd like to now invite our first speaker, Dr. Meng Yao, who will be talking about her work on snow leopards and other mountain carnivores in the Tibetan Plateau. What's fascinating is that she's applying fecal DNA metabarcoding, now I got the right term, to reconstruct feeding habits and niche relationships among mammalian carnivores and aquatic eDNA to detect invasive species in biodiversity. I can't wait to hear your talk, Dr. Meng Yao. Please go ahead. If any of you have questions for now, kindly put them in the chat window. It'll be wonderful if you could please keep yourselves on mute while Dr. Meng Yao is speaking, and then afterwards we'll come back and have some discussions. Over to you, Doctor.
Okay, thank you. Hello, everyone. Here's Meng from Beijing now. Thank you for this opportunity to present some of our study in the past few years with the snow leopard network. So our study mainly focusing on actually a community ecology, like how coexisting animals interact and how they coexist. So today I'm going to talk about two studies mainly. One focuses on snow leopards, and the other one is more a general community study showing how multiple carnivores coexist. Okay, so before I start, okay, just a brief introduction. Who I am. I'm Meng Yao. I conducted my bachelor study in China at Peking University, and then moved on to study molecular biology at the University of Michigan, and got PhD there, and then continued my molecular study as a postdoc fellow at University of Michigan Medical School. But then I came back to Peking University and then converted to wildlife ecology, but incorporating some of my background training with ecology study. So in the past few years, we're trying to use molecular techniques to solve some of the very complex interactions in ecosystems, such as in Southwest China, where many different carnivores coexist. Okay, so why we're interested in diets? Diets not only describe how what a species eats, but it actually describe a lot of complex interactions within a community. So the sum of trophic interactions, just feeding interactions, constitute a food web. Okay, So, and the food web structure and its feeding interactions actually have very fundamental ecological characteristics. And understanding these interactions is very important to ecosystem functioning and stability, and to understand the ecosystem processes. So, without a good understanding of how species interacts in the ecosystem, can't really have a science-based conservation or management design. So that's why we're trying to get a very good understanding of how species eat each other in the ecosystem. From outside, study a species diet seems a very simple question. But actually, a simple question is equal to an easy question. In some ecosystems,
Meng, I'm sorry, Meng. Are you going to share your screen with us? Because that's not shared as yet.
It's not shared?
No. Sorry.
I thought I was sharing. Oh, maybe because then just, oh, sorry.
No problem.
I did click the wrong button, I think. Hold on. Let's see. Sorry about that. Probably because
no, that was our mistake. I think Kaustubh was sharing the screen.
Yeah, I thought it was sharing, and I was playing the.
It's fine now.
Now?
Thank you.
Yeah. Yes, we see it now.
Sorry about that.
Thank you.
Sorry about that. Okay. This is just quickly going through all these slides already. So, okay. So let's see. Okay. So I'm on this right. Okay. So I'm talking about studying diets actually not so easy. In some ecosystems, you can actually see animals interacting, animals feeding, studying diet maybe relatively easy. So in such as in Africa savanna, you can actually see a predator hunting a prey. But in most ecosystems, you don't have that. You don't have that kind of condition, and you don't have that many animals. The animal densities are low, so you don't get to observe these behaviors. Traditionally, ecologists have been using morphology-based method to study diets. So these morphology-based methods, basically we use field collected samples. These samples are fecal samples. You collected the samples of your interested animal, your subject of study, and then you look under microscope to see what's inside those samples. So hopefully you have some undigested hard parts like a feather, a hair, or a claw, a beak, something like that. And based on your good knowledge of species morphology, you identify them back to the original food species. Okay, so just some random pictures here, but you get an idea. The problem was this kind of method. Well, the method this kind of methods have been used for a very long time and generated many very useful, data. But there are a problem. There are issues with this kind of methods. First of all, you base this species identification on morphology characters. But if you have multiple species, very similar with very similar morphology characteristics in the same region, then it's very difficult to do the species level identification. And sometimes you just risk of identifying a wrong species. And also it's based on researchers' experience. So it's kind of subjective. So one person's identification result can be quite different from another person's. This kind of methods can only work for food with hard remains, with a feather, a hair, or something like that. But if you consume something like very soft parts, like intestines or muscle, you don't get any signal. You don't get any trace of food. But you can based on your identification. So that kind of food get no information in your analysis. and also very time consuming, to do this. You have to do sorting through all the individual hard parts in feces. Okay, so that's that. Okay, so for us molecular ecologists, feces actually contain a lot of different information other than the hard parts. Feces contains predator DNA, which you can use for species identification. This for identifying what species generated the feces, and it can do genetics, as just in the introduction. You can do population genetics, things like that. And it contains pre-DNA, the food DNA. So you can do dietary analysis, as I'm going to talk about in a minute, using that DNA information. And feces contains gut microbiome DNA, which you can use, because gut microbiome now is a very hot topic, very hot research area. A lot of human studies are based on this kind of DNA information. And also has parasite DNA, which you can use to study disease transmission, things like that. So very, very useful. So very valuable samples to us. Okay, so and the basic technique we use to identify this prey and predator species is called DNA barcoding. You may have heard a word that refers to short, standardized, well-characterized DNA sequences that you can sequence and to base species identification on. So we now have a big databases, saving all this information. Then you can sequence your sample and then compare with these databases and to see what kind of species your sequence came from. Okay. And now we have an even more complex technique called meta barcoding, which means you identify your mixed DNA pool. It may contains a lot of different species. You amplify the DNA pool with we call general primers. Primers are these short DNA. You can just target a whole group of animals. Say I'm using a vertebrate primers. Like these primers can amplify most vertebrate species DNA. So in this way, you can amplify a whole community of DNA, then you can do a whole community identification. That way you can just identify a lot of different mixed species. Okay, so if I get that clear, so the dietary analysis is based on this DNA meta barcoding technique. So the basic workflow includes 4 parts: sampling, which means you collect feces in the field, and then in our lab we extract the DNA from the feces, and then we process them, amplify them, make the signal bigger, and tag them with different sequence tags, and then we sequence them on this second generation sequencing machine, which is a very high throughput technique to generate tons of sequences at a very short time. And then finally, we'll use bioinformatics to sort these sequences and to assign each sequence to a unique species. So in this way, we can do a lot of sample analysis in a relatively short time and in a relatively standardized way without human judgment involved. So it's we think it's more accurate taxonomic assignment, and it doesn't require much information about a diet before you conduct this kind of study. And you can do this for all kinds of food, including soft food, any kind of animal parts you can detect because it has DNA in it. It's very sensitive and very efficient. So these are very good, for our large-scale diet analysis. Okay, so that's the technical part. It's done. Okay, so now let's get back to our subject of study, the small leopard. So you probably know a lot more than I do about the species. It's we call the apex species or top predator of its ecosystem. And then the whole population, about a few thousand, probably over half, are living in China. So studying their ecology and their dietary habits in China actually very important for conservation of the species as a whole. So we carried out our study in Qinghai, in Sanjiangyuan region. Qinghai is actually very central to snow leopard's distribution range in China. And we summarized the snow leopard diets from different parts of the world to see if if they have a dietary difference by region, and there are significant differences in snow leopard diets. If we look at these numbers, these showing the ratio, although they sometimes the diet in different way, but relatively it's the frequency of occurrence or the ratio of their occurrence in diets. So they're showing like several main categories: the wild ungulates, small mammals, birds, livestock, plant. So in and each line here showing a different region. So we see in Pakistan, Nepal, and India and Mongolia, different regions. There are some patterns as ungulates, either wild or livestock, actually accounted for a major part of snow leopard diets. That's a common phenomenon. And then supplemented by small mammals and birds and other food. Interesting, in China, there are several studies, actually three. Oh, sorry, let's get back. There's 3 studies conducted in Qinghai before. So let's see. Somehow I get this. So the first one was done by Schaller in the 1980s. And actually he found some very interesting pattern in that he actually reported a very relatively small portion of wild ungulates in snow leopard diets, but a relatively large proportion of the small mammals. This pattern also repeated by Liu Chuguang in 2003, showing very similar numbers of these 2 categories. But these numbers are different from a later study done by Li Juan, Lu Zhi's PhD student. She found actually a larger proportion of wild ungulates, but a relatively small portions of small mammals in snow leopard diets in Qinghai. So which one is correct? I mean, is snow leopard diet in Qinghai is different from other parts of the snow leopard range? Are they really consuming more small mammals in Qinghai? But if you notice, all these studies are done by morphology. This actually means at the first stage of identification, which is the field sample collection, they base their sample identification on fecal morphology, which means they see a feces in the field, they judge or they identify its the snow leopard feces based on its shape or color or look. This kind of technique, the morphology-based technique, can have very large mistake ratio if you actually examine them again using molecular-based technique, because the appearance of the feces in the field, they can change with time, or with the weather condition. So if you mistake, say, a red fox or a leopard cat's sample for a snow leopard sample, then that will bias your identification results towards small mammals, because those smaller carnivores consume more small mammals. You can, really get a wrong results. So is that really what happened with Schaller and the Liu Chuguang's result? So we want to find that out. To do that, we collect samples, carnivore samples throughout Senjiangyuan region. We actually collected 7 different sites covering the most of the Senjiangyuan sites. So this is where it's in the bigger map. And these are the 7 sites, and these gray dots are our sample locations. We actually identified each of the samples using the molecular technique to confirm they're actually from snow leopards. So that's, tell us we really get snow leopard sample and the snow leopard diets. So we analyzed altogether 351 snow leopard samples and find 20 different prey species in their diets. Okay, so this is the list of the species, the common name and Latin name. And then here showing you in all these samples the percentage of sequence abundance. We use a word called RRA. It means just relative reads abundance. It's just a sequence percentage in their diets. It's roughly correlate with biomass, but not so accurately, but relatively. You can see here most of the food category. The main food category is this blue colored several species. These are the wild ungulates. It's overall account for 84% in snow leopard diets. So this is consistent with most other regions, snow leopard diets in most other regions. It's ungulates actually account for most of the snow leopard's food. And then livestock, domestic ungulates account for 6% on average in all samples. And then small mammals only like less than 9%, but then a little bit birds. So this result is more consistent with Li Juan's results, but different from Schaller and the other paper. So this is showing probably, I suspect they probably misidentified some other carnivores fecal samples as snow leopard samples. Okay, so here showing you by site, these different sites, the proportion of these different food, in also again in sequence percentage in these different regions. So you can see there are some regional variations. To sum up, there's 20 different taxa. This is total 20, but in different areas, in different sites, they may have more or less. Less than this 20, maybe just a subset of this 20 species. And number of prey, about 1. Most only have 1. Very few have maybe more prey per species. And then we want to say the wild ungulates, actually the whole blue bar here is all blue sheep, or called barrel, and then several other wild ungulates here, but very small percentage. So 80% is about for this 1 species. And then domestic yak is the 1 domestic livestock that's consumed the most. And then marmot, it's a small mammal that's got consumed also. So here showing the Senjiangyuan area, the total diet composition. And in this map, we show the diet composition in 4 main categories. The blue part is the wild ungulates, and the red is livestock, and then the brown is small mammals, and then a little bit birds you can't really see because it's too little. And you can see this in different areas. The proportions of these different categories vary. They may have some regional differences. So we did some statistics and showing, first of all, no the difference among years, because in some sites we have multiple years samples. So compared among years, no difference. When compared between the warm season and cold season, no difference. And then we conducted a comparison between sites, and there's significant difference, meaning the snow leopard's diet can vary by area. Okay, and then because snow leopards is one of the main carnivores in the region, and some people may think they pose threat to local livestock farming. Because the snow leopard have been observed to prey on livestock. So we analyzed the snow leopard preference for livestock and burrow based on their percentage, their proportion in diets, and also their availability in the field. This availability data actually are based on camera trap records of these different species. So it's a measure of the relative to their availability, how much snow leopard consume each one category, and this shows if snow leopards prefers or avoid this kind of food. So by doing this, just to sum up, it's kind of complex. So different sites, the 7 sites, and in all these sites, the red showing avoidance and the blue showing preference. So in all these sites, snow leopards strongly prefer wild prey, which is burrow, relative to livestock. And we found only when livestock density was substantially greater than burrow density, snow leopard will increase their livestock consumption. Okay. So we further analyze what factors affect snow leopard's livestock predation. So here we're showing these different correlations between livestock consumption. This RA showing is the proportion in snow leopard's diet. This livestock consumption versus different ecological factors. And in all these different comparisons, we found that this one, livestock biomass has a positive, significant positive correlation with livestock consumption. While other factors were tested, livestock density, burrow density, or the relative density of livestock versus burrow or snow leopard density has no correlation with livestock consumption. So this is very interesting as because livestock density is usually used as a metric for measuring, the snow leopard selectivity for livestock. But here we're showing biomass actually is the factor that actually has this positive correlation. We think this probably has something to do with the way livestock distribute, across landscape, because it's not a really uniform distribution, but more a heterogeneous and more patchy distribution, very clustered distribution. So a larger flock of livestock may attract more attack from wild carnivores. That's some hypothesis we have. Okay, and the last one we also factored in a snow leopard density with this livestock. This so gave a measure of the total consumption of the livestock in snow leopard diet. So this also showing very a high significant positive correlation with livestock biomass. So this is just showing overall livestock loss to snow leopards is positively correlated with livestock abundance or biomass. So next, we also analyzed some other factors, the factors affecting snow leopard density. If we look at graph panel A through D, the y-axis are all snow leopard density, and we correlated with the burrow density and livestock biomass. And the other one showing a positive correlation is with the burrow density. So this two actually are similar, but this is with 6 sites. This on the verge of being significant, but we include another different site. So this one is highly it's significant. So showing snow leopard density is positively correlated with bird density, which means you have more wild prey, you have more when you have more wild prey, you have more snow leopard. So this is very important for conservation, right? So you know the wild prey recovery maybe is the most critical factor for snow leopard conservation. And other all being insignificant, but we see a negative trend between livestock biomass and burrow density and the livestock density with burrow density. So this may suggest that increased livestock has a negative effect on wild prey. So, you know, so limiting the livestock abundance may be also important for the conservation of wild prey base. Okay. Okay, so there are actually 2 different mechanisms suggested for prey interactions for two prey sharing one predator. Their interaction may follow 2 different patterns. One is called apparent facilitation, which means increase consumption in one prey can reduce the consumption in the other prey, which is actually pretty straightforward because one predator is this one more than is this one less. So this is called apparent facilitation. But the other mechanism called apparent competition, this works with this increased consumption of one prey actually results in increased population of this predator, then in turn increased consumption of the other prey. So both up arrow. So this means if you get more wild prey, then you get more snow leopard, and maybe more livestock can be consumed. So both mechanisms or both patterns have been observed with snow leopards in different regions. But in Sanjiangyuan, we think that this left one may, or at least more similar to the patterns we observed. Because we don't actually see an increased prey results in more livestock. We actually observe more wild prey resulting in less livestock consumption. Okay. All right, to sum up this first story, to be quick. So snow leopards actually prefer wild prey relative to livestock. Livestock loss to snow leopards positively correlated with local livestock abundance or biomass. And the snow leopard population size is mostly critically dependent on wild prey abundance. Okay, so the implication for conservation biology, it's first, wild gum-wooded population recovery is key for snow leopard population growth. And also, we need to really control livestock total abundance, but then for those already, livestock already in place, you really need to improve your guarding practices to prevent, to minimize losses to snow leopards and other wild carnivores in the region. Okay, so this is our first story about snow leopards mainly. But we are still analyzing this data because we also collected other carnivores feces together with snow leopard samples. So we're doing analysis on those samples to see how these different carnivores interact. But we have already done that for different ecosystems. So I'm gonna talk briefly about that one, hopefully in time. So we actually, not snow leopard, but overall, all large carnivore populations are declining around the world. But there are a few regions that still have a rich carnivore species diversity. One of them is in Southwest China, which on the east edge of Tibetan Plateau. So this region actually have very high carnivore species richness. Sanjiangyuan is actually within a general range of this region. So we collected samples from other ecosystems in this range, including Minshan, Qionglai-shan, and Shaolishan. These are just 3 different mountains in this general region to see, what difference or what similar patterns they have with their carnivore communities. So this region has a more diverse habitat types, landscape than Sanjiangyuan. So it may offer very diverse conditions for different for different carnivores to coexist. So we start our sample collection. So this is me with my colleague Sheng, Professor Li Sheng, and this my students collecting samples in the region. So we see a lot of different feces in the field. The first step is to determine whose feces they're from. So we use the molecular technique, as I said before, to identify the predator species or the consumer species at first. And so these are just different figures showing the typical sample of these species look like. Okay, so together we collected in these 3 different regions, collected 1,700 fecal samples. We did a species ID, identified 1,400 feces, and then these are from carnivores, and then we did a dietary analysis, and 900-ish samples had very good data for our further analysis. So using these samples, we identified these communities at each of these sites. So this is the Minshan site. They only have 2 smaller carnivores, leopard cat and the golden cat. Then Qionglai-shan in the middle has 3, 2 smaller ones, and then snow leopard, this middle. And Shaolishan is the most diverse system and has 2 smaller carnivores and then 5 large carnivores, including 4 wild ones and then 1 domestic dog, because there's a pastoral community and has dogs around household. Okay, so these are different systems, have a different carnivore communities. So we first analyzed the dietary composition for each of these species in each of these ecosystems. So Minshan has leopard cat, golden cat. You see these, together we found 95 pre-taxa, or most of them to species level, but some at a higher level. These include 64 mammals, birds, and then some fish and reptiles. So here I can't really show you individual pre-species. So here we're showing by order, like different order food in their diets. So the general pattern is a smaller carnivores like leopard cat and the golden cat. They have large proportions. This grayish color showing this is the rodents in their diets. This is the same in all 3 systems. And also this green part is the lagomorph, like hare and the pecas and stuff. So these are their main prey. And then for large carnivores like a snow leopard, you see this big green bar. This is the here. It's the ungulate. Okay. And also same here, the leopard is the green part is the dominant part. These are the wild ungulates. And also for large carnivores in the Shaolishan region, you see this very big parts, the yellow color. This is livestock. So in Shaolishan, they not only have wild ungulates, but also a lot of domestic ungulates in large carnivore diets. Okay, so to see interspecific interactions, we constructed food webs in these ecosystems. And in these graphs showing on the top are the predators and the bottom each bar it's prey. So you can see with more carnivores, you have more complex interactions, all these lines creasing, crossing each other. So this is just showing you how complex a food web can be. And this can only be resolved using the molecular technique. So just to blow up this complex system to show you some of the main predator and prey. Here, this is the Shaolishan ecosystem. You're seeing all these carnivores on top, and each one of them is prey. There's 59 different prey category, and then can show you each one of them, but some of the main species are here. So just they have a lot of interspecific overlaps in their diets. Actually, I did a lot of the quantitative analysis of their niche overlap, the dietary niche overlap. And here showing you a graph of carnivore body size in relation with their prey diversity, and a lot of dots. But anyway, take home message is the prey diversity actually decrease with increasing carnivore body size, which means smaller carnivores like leopard cats and the red fox, they actually consume a lot more or more diverse prey than large carnivores. Also, we did a correlation between carnivore body size and their prey body size. This actually factored in the proportion of each different prey species in this calculation. But anyway, we show a very good linear correlation between carnivore body size with their prey. This indicates, larger carnivores actually prey on larger food species. Right, this is also it had been suggested in our ecology books, but actually show empirical data at this kind of level. It's actually very rare. And actually there are some details to point out, like wolf and dogs, they're actually above this linear correlation. We think probably because their group hunting strategies can help them to prey on or to get larger prey, larger than predicted on their individual body size. Well, brown bear is below this line. Brown bear is a very opportunistic feeder. They actually eat a lot of smaller prey like marmots. So maybe account for their lower than expectation. So anyway, just some trends with their dietary composition. And also did the prey preference of large carnivores in Shaolishan. And we show this, just look at the bottom graph. Okay, so each line is a different large carnivore species, and vertically these columns showing different wild ungulates and domestic ungulates. The last three are domestic ungulates. Okay, you see, except leopards. Leopards show a very strong preference for a wild prey. This is a Chinese squirrel, but avoidance for all other species. Well, deeds, wolf, dog, and brown bear all showing a high preference for domestic prey, the livestock, showing by the blue, but avoidance of most wild ungulates. So this suggests in Shaolishan area, domestic, the livestock may have become a very important substitute food source for large carnivores, as wild prey availability, is reducing because of human activity. Okay, so yes, okay. Okay, to sum up, behind my time. So here showing this image summarizing our key findings in these three different ecosystems. So you have a diverse prey base to support your diverse carnivores. But then the large carnivores and the smaller carnivores, I will call mesocarnivores, they also separate, or they also show some dietary partition, which may facilitate their coexistence. Like smaller carnivores may consume more smaller prey like small mammals and the birds. But large carnivores mainly consume ungulates, either wild or livestock. And in Tibetan areas, because these cultural tolerance for livestock consumption, maybe large carnivores can subsist on a lot of the livestock in this region. So this is also very important to know for their conservation in different regions. Okay. Alrighty. So hopefully I have convinced you this is a very important or a very powerful technique, the DNA metabarcoding dietary analysis. When it's properly integrated into our study, into our ecological question, we can actually address very complex, very intricate, interspecific relations. Okay. All right, so thank many people. First of all, thank my students who have done hard work. Qi Lu and Xining Shao, who conducted the main experiments for these studies, and also with help of many other students. And thank my collaborators, Dr. Lu Zhi, Li Sheng, and Da Jun, all gave me a lot of help. Thanks Shen Shui and CC actually helped organizing many samples. And thank the nature reserve staff helped us collecting some of the samples, and the volunteers also. And thank my funding source. And thank you for joining us on this webinar, and I'm ready to take questions. Thank you.
Thank you, Dr. Meng Yao. What a fascinating talk. It's almost as if you shared with us an encyclopedia of knowledge with what you've been able to collect. Thank you. No, absolutely. I'm sure, and I'll take the I'll take the opportunity here to request our participants, if they're okay, that we extend the meeting for a few more minutes, because I'm sure there are hundreds of questions already. And before we move to those questions, I'd like to now invite Dr. Cheng Chen. She's the program director of the Nature Watch program in the Shanshui Conservation Center, CC as we all dearly call her. I'd like to request her to please share her perspectives from the wider work on snow leopard genetics in China. Dr. Cheng Chen.
Hi, can you hear me?
Mm-hmm. Yeah,
great. Thanks for Meng to give such a fascinating presentation, and I was very honored to be part of very little part of this research. So I'm really glad to see this published, and today we have the webinar to share the results. It's very exciting. And actually, I think maybe metabarcoding is very new even in genetics. So I don't know if everybody just get the enough information or insights from the talk that metabarcoding, how it works. But I want to ask Meng a question about is this technique, do you think this technique is currently ready to be widely applied in conservation work? For example, is it easy to collect qualified sample by conservationists or some NGOs or some local people? And is it difficult or technically mature to achieve this genetic data and dieting results? Can you share some more information with us?
Mm-hmm. I think the sample collection part is easy. You just need to be careful about cross-contamination. You just handle each sample individually with clean gloves and tubes and stuff like that. It's not a problem. And then we can preserve samples in the field with easy ethanol immersion or with some silicone beads to keep it dry. That's not difficult. But for the analysis part, you do need a molecular lab and a qualified one to make sure your good data quality, because it's you use some of the techniques very sensitive to contamination, like PCR, like high throughput sequencing, like metabolic coding. These are very sensitive to just a little bit DNA mixed in, you can get a very strong signal. So we are very careful. We include a lot of negative controls to monitor the potential contaminations at each step. So just to make sure, because there are a lot of steps, and at each step you may have some risk of cross-contamination. So you need to have a very strict workflow, and also data processing, and you have to be very careful. So, but this comes with experience. The technique itself is not that hard. I mean, it's just basic PCR and stuff like that. But just we are sensitive to contamination, which you need to be very careful, because once you mix, say, you mix a snow leopard sample, if it touched on a leopard cat sample, then maybe some of the leopard cat DNA can transfer to the snow leopard sample. Then you can have a lot of small rodent signal or stuff like that in your results. So that's something that can mess up your the quality of your data. So yes, so yeah, for the first part, it's not that difficult, but then for the second part, analysis, the lab analysis and data analysis, you need to have very good quality control, for those parts. Yeah, so I think it's best to work together, because you can work with nature reserve staff and to train them to collect samples. That's not easy. We have done that. It's very easy. They follow protocol, wear gloves and put in a tube, tighten up, and then put in fridge, or you put in some ethanol. That's fine. Then they transfer back to the lab, and then we take over from there. So yeah, I think a lot of I've seen more and more studies from a lot of more countries now, not just the very technical advanced countries, but also from the snow leopard range countries. So I believe it's doable, and it's just with good quality control, with good practice of laboratory techniques, you can get good results. Yeah, just be careful. Yeah.
Thank you. Also, I think, yes, maybe everybody knows that there are still a lot of challenges in the technical measures. So by now, I think this technique is ready for research, but not not totally ready for application, widely application. So maybe we we just find a qualified lab and experienced team to do some collaboration to get those results that we need, right?
I think that's a good idea. If you have zero experience with this kind of technique, rather than establish your own lab, get everything ready, probably better ways to find a qualified lab to collaborate. Can save a lot of trouble.
Yes, I totally agree with that. And according to this interesting results, I noticed that you mentioned that actually it's a good news for me that in Sanjiangyuan, at least during your study period, snow leopard is not the main cause of livestock loss. Yeah, but compared to the other researchers in other areas, maybe they have a very different finding. So what do you think what the reason do you think causes this difference? And I'm also very curious about what happened in other areas. So maybe we can just open up a discussion. I don't know if this webinar allows the other listeners to speak out. I just have these two questions, and maybe we just discuss about this interesting story.
Yeah, shoot story. I mean, I'm not a trained snow leopard ecologist, so from my experience and my reading, I think a lot of what snow leopards eat have things to do with their prey base. What are available at the ground, in the local ecosystem. And I talked to Ling Yunxiao, who is also another PhD student of Lu Zhi. She also said something about high availability of local wild prey may explain partly, at least, for the mechanism between the wild prey and livestock in snow leopards' diets. Because you have a very low wild prey already, then any increase may just increase snow leopard density, then result in more predation on livestock. Then you get a different mechanism. You may have a negative effect from increased wild prey. So there's very complex interactions. We can't say what happens now is always this way in Shenyang and always will be in the future, but currently it doesn't look like it has a negative impact. So we still think increasing wild prey availability will be a very good thing to do, both for snow leopard conservation and also for local livestock protection, because snow leopard can rely more on the wild prey burrow.
Thank you. And I think maybe Sanjiangyuan actually, from my experiences in working in Sanjiangyuan, it's a paradise for wildlife, because the human population is not that it is not very big, and the development is very limited in this area, and the policies have actually cut off a lot of livestock in the history of Sanjiangyuan. So maybe your finding represents a very healthy ecosystem or food chain, that the snow leopard have the choice. Because I have in my field work, I used to hear that the local people said snow leopard doesn't like the sheep or the small livestock, because they think they are too small and they are not healthy. They are not as healthy as blue sheep. If snow leopard eats too many blue sheep, they will be not as healthy as they are currently. So yeah, that's a joke. But I think maybe if they have a choice, a snow leopard may prefer bigger prey, so that they can hunt for once and then be for longer. And also they want more healthier and stronger prey to just keep them strong enough. So I think maybe it's another that it shows how healthy the ecosystem is. Yeah, that's my thinking. And yeah, I'm done. And maybe we can just have others to be in this discussion.
Thank you, Chengchen. That was a really fascinating conversation, and thank you to both of you. We do have a few questions. I'm just going to pick up a couple of them. One of them is from Narayan Koju. I'm going to pick the second part of his question, which is quite interesting. He's saying that he is doing some diet analysis of snow leopard from eastern part of Nepal, and they found hair samples of snow leopards in the snow leopard's cats. Is this common? The only point he mentions here is that they're using histological analysis and not genetic analysis. So would you have something to comment on that one, Dr. Mengia or Dr. Ching-Chen, either of you?
Hair, yes, yeah, we do see, because the scan analysis before the did before, like Shala and Liu Chuguang and Li Juan, they did based on these morphological characters of the food remains, just such as hair. It's a very important source for their species identification. So definitely hair, and also found a lot of plants like twigs, but we didn't do any plant analysis, so that's surprising. Hair, I think is normal.
So I think hair of snow leopard itself, that's what their question was, which is common.
They always know like hair balls. Yeah,
sure, makes sense. Makes sense. Okay, there are a few appreciation comments. Philip Kirch has expressed his gratitude to be able to attend this talk. Okay, there's one more question. I think that's from Justine. How many yak normally in a herd or clumped together on an average?
That I didn't have a good answer for that.
You mean Sanjiangyuan?
I think pretty big.
I think dozens to 300 to 400. Yeah, because I think it's different in different areas in Sanjiangyuan. Is when you go to on-site, one of the sampling sites that Meng has mentioned, they don't live on livestock. On-site has a very good caterpillar fungus. So so, and also the policy doesn't encourage the livestock grazing. So now they have maybe just dozens, 60 to 70 for one family. But in some other areas, like some people have even rented people from other areas to come here and stay here to raise the livestock for them. So in that case, some of the families has 300 to 400 livestock. So it depends.
Yeah, fantastic. In Shaolu Lishan, they also have yak like flocks, but it's really varies. It's very by household, and even they have a big number of animals. They can, go in smaller flocks. And actually, I found they don't really attend their animals. They just leave them out in the mountains.
Yeah, especially males.
Yeah, for months. For months. They don't even know how many will come back.
Actually, they are not good at counting. If you ask,
no, they don't have.
Do you know how many? They just say 100 or 200.
Right.
Yeah, in fact, if I'm not mistaken, that to a large extent is also accredited to be one of the reasons why such large-bodied prey are more in the snow leopard diet than the smaller bodied prey, which are more herded and more proactively guarded. So fascinating. Okay, so I have a couple of questions, if you guys allow me to. And one of them is, I think it's about the variation that you showed that was quite fascinating. Have you tried or have you considered doing individual ID of the snow leopards whose scats you're analyzing? Because the only reason I mentioned this is from some other studies that I've been privy to, especially let's say there's one on tigers where 7 different individuals had different skill sets of learning to kill certain prey. So one tiger was specialized in killing an antelope. The other tiger was specializing in killing the cervus species, and so on. So each of them had their own specializations, and their cats had more of those. So have you considered doing individual ID and looking at that as a possible factor behind some of the variation that you're observing?
Right. Well, that's a very interesting point. I have read some of the studies showing, the mother son, feeding habits, teaching, stuff like that. No, we haven't done that in a short answer. It's much more resource intense to do that. You need a lot more molecular markers to do individual identification. A lot more work. Yes.
So maybe a related question would be that when you collect these cats, we're hoping that in the next few months the snow leopard genome or the SNPs might be ready. And apparently, if I understand, whatever little I understand, they say that it's going to be easier, more easily comparable, and more standardized. I think those processes, cheaper, maybe cheaper as well. So can these samples that you already have be used for genomic analysis as well? Will it be possible?
I think so.
Should
We still keep them frozen with all the DNA hopefully intact. We don't know how long it lasts. Yeah, but it's definitely more. Still need a lot of money to do that. Even they have SNPs or whatever. Yeah, the SNPs you need more markers than microsatellites, many more. If you have chip or something like that, then that will cost a lot also for individual identification. So if you have a proper question, if you have a very good research hypothesis to test, I think it's worth it, to do like kin relationship or to do sex ratio or do things like that. It may be worth it to do that. Yeah, it's beyond a little bit beyond what we
I understand
now at this stage. Yeah.
Fantastic. No, but this is already very fascinating, and the relationships are quite intriguing. And there's no end to how much you can research, right? And how many more questions you can ask. So yes, someday hopefully, if there is this individual choice between individuals, that could be a question to answer as well. Fantastic. Great. So that's wonderful. There's no more questions, so I'll go back to my second question, which is you mentioned a lot, and that's been a challenge with a lot of older studies, that we assume a scat is snow leopard scat, and then you analyze many times. You might be analyzing a fox scat, many times you might be analyzing dog or wolf scat. In fact, there's a study in Kyrgyzstan where the researchers found that not only 67% of the scats that they thought were snow leopard turned out to be something else, mostly wolves, I think, but 22% of the scats that they thought of were of wolf turned out to be snow leopard. So there was like error on both sides. And I was just wondering, I haven't seen your paper, so my apologies in advance, but have you mentioned these proportions of how much can it get wrong? Because these are important numbers that people need to realize when out in the field, and they believe this is a snow leopard scat, but many times it's not. So any metrics on that front?
We did some of the analysis, but because in our analysis we're collecting all putative carnivores scats in the field, so not always
makes sense.
Identification are given in the field. So people are just collecting and not really judging what species they are. So we don't have that kind of information.
Makes sense.
We did some, and yes, the mistaken rate is very high. And I think I saw a paper like summarizing the mistaken rate, about 40-ish percent mistake are made even just at the first stage of like fecal identification. So that's, if you think about all the following mistakes you can make, then the final results, you probably get a lot of false signals. Yeah. So
yeah,
Excellent.
I can also share our data because
Mhm.
we in the field when we do sampling, we always make a record that if you think in the field that this is a snow leopard scat, so that we can compare when we get the species identification molecular by molecular data. So for us, even for our most experienced samplers like me, there are always 30 to 40% that you make mistake. Yeah, because
Wow.
if you, I don't think it's please don't feel uncomfortable, but if you just look at your own scat, you will see it the shape and its looking changes every day, depends on what you eat. So it's the same for snow leopard. When they just eat something that is not so good, they will just give it something that is not identifiable. Just give up something like that. And if they just eat some plants, that will be a very different shape from their just a very common like a ping pong ball scat. But actually, when we are doing sampling in the field, if we see this kind of scat with such plants inside, it will be more, we are sure it's from snow leopard because other carnivores don't eat plants like that. So actually that kind of scat just can raise up our current rate. But for other side, if you just look for some very snow leopard scat looking scat, that may make mistakes because wolf can also sometimes they also just have this kind of scat, and snow leopard sometimes do something different.
Excellent. Thank you, Cheng Chen. That's very yeah. You explained it very well. I'm sure nobody will have any doubts with that easy to understand explanation about how scats could vary. Thank you very much. Here's another question from Dr. Justine Shanti Alexander. She's asking, what are the future questions Dr. Meng Yao's team is interested in looking at? What's there in store for you in the future?
We are actually looking at other carnivores coexisting with snow leopards on the Tibetan Plateau in the Senjiangyuan area to see, because there are also many other animals, many other carnivores together with snow leopards, such as gray wolf and brown bear and the lynx and the Tibetan fox, red fox, leopard cats. So there are many more. We want to see if they're, because Senjiangyuan ecosystems relatively simpler than the second story I told you about in Sichuan area. The prey base are more diverse in the Sichuan, in the Three Mountain area what I just talked about. So in a relatively low productivity region like Senjiangyuan, how all these carnivores coexist. They may have stronger competition, or they have stronger dietary separation. It could be a different pattern. It could similar. We don't know, because I haven't seen much, dietary data from these high elevation areas in the world. Most what we know about carnivore community coexistence comes from Africa, comes from South India, these areas with very high primary productivity and high prey diversity. So I want to see if very similar, in a similar, pattern or a different pattern mechanism can work at the highest part of the world. Yeah. And also we are doing different from these. So what we have from what we get from scats is called the eDNA. It's environmental DNA. It's a type of environmental DNA. It's just from physics. It's a very unique environment. But we can actually get DNA from water, from soil, even from air to study wildlife. So we're actually doing that on the Tibetan Plateau, also in many different systems, in cities and also in other regions around China to see what species we can sample in water to see what wildlife we can, to catch by just collecting some water from rivers and lakes. And it's a different live study, but also very interesting. I hope, but no snow leopard now. But potentially, if we do it on the Tibetan Plateau, say in a region where different wildlife can drink or go drinking in a pond or a pool, something like that, we can also sample wildlife in that way. So it's also very fascinating new area of study. So that's something we're doing. Yeah.
Thank you. Just before we leave, one more question just dropped in. Have you found any fish in the snow leopard diet?
I don't think it's snow leopard diet. No.
Okay.
Not
Interesting.
But okay. two birds, no fish.
Of course. All right. Thank you. All right. Yes, I think with that, really, I'd like to first and foremost thank both our speakers today, Dr. Meng Yao and Dr. Cheng Chen, for providing us, a window into the new developments, especially in the fecal DNA metabarcoding, and just showing us what all is possible. And especially to you, Dr. Meng Yao, the way you showed the way the species are interacting, the way the animals, their diets are varying. It was absolutely fascinating. And given that conservation genetics is a rapidly evolving field, I think today's talk was quite valuable in many more ways than one. So really, thank you one more time, and see you all soon in another meeting of this sort. Thank you. Have a good day. Have a good night, and take care.
Thank you.
Bye-bye.
Thank you. Thank you. Thank you all.